Merhaba. Sayın hocalarım, sevgili öğrenciler, herkese merhaba. Bir sürekli tıp eğitimi toplantısıyla yine birlikteyiz. Ve bugün çok değerli bir konuğumuz var. Ve
Merhaba.
değerli konuğumuzla geleceğin ilaçlarında
Hocalarım, sevgili
nanobiyoteknolojinin
öğrenciler,
etkisini
herkese merhaba.
konuşacağız.
Bir
Evet,
sürekli
bugün
toplantıyla yine birlikteyiz. Ve bugün çok değerli bir konu.
bugün konuğumuz Doktor Şeykur Musa ve biz Abdelati ile birlikte bu programın moderatörlüğünü birlikte üstleniyoruz. Ve bugünkü bu iki değerli bilim insanının akademik özgeçmişi hakkında kısa bir bilgilendirme yapmak istiyorum. Profesör Doktor Şeykur Musa, New York Eyaleti Üniversitesi'ne bağlı Albany Üniversitesi'nden farmakoloji profesörü. Ohio Eyalet Üniversitesi'nde farmakoloji doktorasını, Kentucky Üniversitesi'nde doktora sonrası araştırmalarını tamamladı ve Pennsylvania'da Widener Üniversitesi'nde MBA derecesini aldı. Profesör Doktor Musa 20 yılı aşkın bir süredir New York Albany Üniversitesi'ndeki Farmasötik Araştırma Enstitüsü'nün kurucusu ve başkanıdır. Çok sayıda yardımcı ilaç ve biyoteknoloji şirketinin kurucusu, yönetim kurulu üyesi ve başkanlığını yürütmekte. 20 yıl boyunca kıdemli baş araştırmacı ve araştırma görevlisi olarak görev yaptı. Temple Üniversitesi, John Hopkins Üniversitesi, Buffalo Albany'deki New York Eyalet Üniversitesi, Rensselaer Politeknik Enstitüsü, Sherry Trombos Araştırma Merkezi tıp bölümünde tıp alanında akademik görevlerde bulunmuştur. 2017 Kuveyt Bilimi Geliştirme Vakfı Tıpta Uygulamalı Bilimler Ödülü'ne layık görüldü. 2018 yılında Ulusal Mucitler Akademisi üyesi olarak ödüllendirilen Musa, 2020 yılında insan sağlığının geliştirilmesi ve iyileştirilmesi için geleneksel tamamlayıcı ve alternatif tıpta kehanet ilaçları alanında ilk Şeyh Zayed Uluslararası Ödülü'ne layık görüldü. Doktor Musa Stanford Üniversitesi'nin 2020-23 yılları için derlediği listeye göre kendi alanındaki bilim insanlarının ilk %1'i içinde. 38 bin üzerinde makalede kendisine atıf yapılmıştır. Mevcut H indeksi 100 olup araştırma katkılarının etkisini ve önemini yansıtmakta. Doktor Musa'nın profesyonel başarıları arasında çeşitli patentler ve yeni antitrombotik tedaviler, non-invazif miyokardiyal perfüzyon ve trombus görüntüleme ajanlarının keşfi ve geliştirilmesine yaptığı katkılarda yer almakta. Binden fazla hakemli dergide yayınlanan çalışmaları mevcut. Yine 400'den fazla ABD patenti ve uluslararası patente sahip. Nörofarmakoloji alanında da çalışmaları mevcut ve bunlar ağırlıklı olarak ağır modülasyonunda hormonal ve nöronal faktörler arasındaki biyokimyasal ve farmakolojik etkileşimi içeriyor. Birçok FDA onaylı antitrombotik ajanların keşfedilmesi ve geliştirilmesine katkıda bulunmuştur. Yine hedefe yönelik tümör radyoterapisi ve görüntülemesinin keşfinde, aynı zamanda heparinlerin ve heparin türevlerinin yeni farmakolojik yönlerinin keşfinde de rol almaktadır. Yine anjiyogenezde integrin alfa 5 beta 1'in rolü, anjiyogenezde kinojen gibi fibrinotik bileşenlerin rolü de dahil olmak üzere birçok temel kavramın geliştirilmesine katkıda bulunmuştur. İlaç keşfi ve ilaç geliştirme alanlarında araştırmalarına odaklanan son teknolojiye sahip Farmasötik Araştırma Enstitüsü'nün kurucusudur. Enstitü, biyoteknoloji, nanoteknoloji, kök hücreler, ilaç tasarımı, yüksek verimli tarama, formülasyon, ilaç dağıtımı, farmakokinetik, farmakodinamik ve farmakogenomik gibi çeşitli etkileştirme teknolojilerinde yararlanmaktadır. Amerikan Kardiyoloji Koleji, Ulusal Klinik Biyokimya Akademisi, Amerikan Kalp Derneği ve Ulusal Mucitler Akademisi olmak üzere birçok ulusal ve uluslararası derneğin üyesidir. sayıda yüksek etkili bilimsel ve tıbbi derginin yayın kurullarında ve hakemliğinde de görev yapmaktadır. Evet, şimdi sevgili Profesör Abdelatin'in de akademik geçmişinden kısaca bahsetmek istiyorum. Profesör Doktor Abdelati farklı ülkelerdeki çeşitli tanınmış kurumlarda prestijli görevlerde bulunmuştur. Mısır'daki Kahire Üniversitesi'nde seçkin bir farmakoloji profesörü ve Çin'deki Kilo Teknoloji Üniversitesi Gıda Bilimi ve Mühendisliği Koleji'nde üst düzey yabancı yetenek profesörü olarak görev yapmıştır. 2013'ten Ocak 2018'e kadar Güney Kore'nin Gimanju kentindeki Honnan Ulusal Üniversitesi'nde beyin havuzu üyesi olarak çalışıyordu. Bu süre zarfında Kore Cumhuriyeti'nde Seul'deki Konkuk Üniversitesi'nde yabancı profesör olarak görev yapmıştır. Halen Ocak 2018'den itibaren Erzurum Atatürk Üniversitesi Tıp Fakültesi Farmakoloji Anabilim Dalı'nda yabancı profesör olarak görev yapmaktadır. Başlıca araştırma alanları gıda bilimi ve teknoloji konusunda olup özellikle çeşitli ekstraksiyon ve analitik yöntemler kullanılarak kısanabiyotik analizine odaklanmaktadır. Yine prestijli dergilerde yayınlanmış 462'den fazla makalesiyle etkileyici bir yayın geçmişine de sahiptir. Mevcut H indeksi 42 olup araştırmalarının katkılarının etkisini ve önemini çok da güzel yansıtmakta. Profesör Abdelati araştırma çabalarının yanı sıra önemli editörlük görevlerine de sahiptir. The Journal Lipid in Health and Disease'in genel yayın yönetmenidir ve son 10 yıldır Journal of Advanced Research'te yönetici editör olarak da görev yapmakta. Yine biyomedikal kromatografi ve ayırma bilimi Plus'ın danışma kurulu üyesidir. Profesör Abdelati dünya çapında uzmanlığıyla tanınmakta ve 2017'den 2027'ye kadar gıda katkı maddeleri ortak uzman listesi uzman komitesinin bir üyesi olarak atanmıştır. Yine Stanford Üniversitesi'nin 2020-23 yılları için derlediği listeye göre kendi alanındaki bilim insanlarının ilk %2'si için. Evet, bu kadar etkileyici, güzel iki akademik geçmişine sahip hocalarımızla birlikte olmaktan mutluyum ve bunları sizlerle tanıştırmış olmaktan da mutluyum. İlk önce konuğumuza hoş geldin demek istiyorum. Welcome to Dr. Musa. We are very happy to have you as our guest today.
So much.
Yes, absolutely. You have to say,
Okay.
please go ahead.
After this comprehensive introduction of civil rights, we are happy to have such a high caliber scientist, outstanding scientists that can merge or combine between academia as well as industrial world. I cannot say any more like what Sevilay already said in Turkish. So we are happy to listen to Professor Shakir for his talk today. So please, Professor Shakir, share your screen and then start your presentation.
Can you see my screen?
Yes, yes.
Yeah, it is. I want to thank you for the introduction in a way, and I appreciate the opportunity to be able to have a discussion with you about the impact of nanobiotechnology on the future of medicine and how this can lead us toward the precision medicine. And I'm going to share with you case studies, but also I like to emphasize that nanotechnology, along with biotechnology, can really help us develop, especially with the case against coronavirus and other infectious disease and many other diseases, can benefit tremendously from that technology. I'm just going to say it up front. Whenever you have an antigen or an antibody, this is a protein. So you cannot really inject it directly into the human being. You have to preserve it and protect it from degradation. You talk about gene therapy, you talk about, antibodies. but any biologics, would actually would benefit from using nanotechnology for delivery. And more importantly, the next step after simple delivery is to target the delivery system into the target itself. If you want to target, certain genes or certain virus or bacteria or cancer cells, so you can use specific molecular signatures and develop a high affinity ligand, and then you can decorate the surface of the nanoshell with those targeted molecules, and then you can encapsulate inside the active therapeutics. And this way you can have a fully targeted therapy that will be more effective and safer. So you don't have to deal with a spillover into other organs that do not need to be exposed to the therapeutics. So in a way, nanotechnology would have, and is proven to have, a major societal impact and economic impact. We realized here, Albany is the capital of the world of nanotechnology, and they use it, in developing a lot of the chips, the cell phone that you have, the wafer, So there is a huge, nanotechnology is not only in the medical field, but in many other, facet of life, in agriculture, in even construction, in medicine, in air pollution, water purification. So you will have nanotechnology being, employed in many other facets in our life. And you can start from point zero here, and within 10 years, 1 decade, and maximum 2 decades, you can reach the full potential from an economic and societal impact of nanotechnology. If you compare this to the Industrial Revolution, took about 200 years plus to realize its full potential. So this is a highly accelerated impact, that can actually, a university or academic or industrial partnership between academia and industry can realize the full potential within 1 decade, which is highly accelerated, in terms of the economic and the societal impact. Why nano? It's, in the medical field, I'm going to just focus on the medical field here, but as I mentioned, nanotechnology has major impact on everything in our life. Nano is, you have a surface area that's very large surface area, and the size is very small in nanometer scale. And it took about 1 nanometer to 200 nanometer scales, which is you have to use, a scanning EM or atomic force microscopy to see the particles. And in the medical field, the traditional medicine that we have, when you take any medicine, it goes everywhere. And, if you are like chemotherapy, for example, it goes, you have a tumor somewhere, but it will go through the whole body. So the patient will have to deal with a lot of side effects and adverse effects. But with the nanotechnology, we can actually target the tumor itself and deliver the drug, the active, the chemotherapy, for example, can be targeted into the tumor with minimizing the spillover into other normal organs like nerve or muscles or liver or other organs that can have a deleterious side effects. So why is that advantage of this nanocarrier? Nanocarrier, I'm talking about pharmaceutical, small molecule. I'm talking about biopharmaceutical, biologics. But, if we think about, pharmaceutical, small molecule drugs, which is the majority of the medication that we have, and even moving forward into biologic, which is large antibodies, for example, or an antigen in the case of vaccine, you can improve solubility, stability, you can specificity, and multimodality efficacy. So this is in a sketch here of a nanoshell. What we can do, we can, encapsulate inside the nanoshell the active therapeutics that we intended to deliver, or even combination, of active therapeutics. And so you can combine, in the HIV, for example, they use multiple drugs. So instead of the patient taking, 5 different drugs, you can encapsulate those 5 drugs inside the nanoshell. And there is an application in that regard. And so you can put say in the case of cancer, you put the anti-cancer drug chemotherapy here. Then you have the surface. You can actually you can do surface modification, surface coating of the nanoshell. You can put an imaging probe, so you can actually image the delivery of the nanoshell and the delivery into the target. You can put a targeting moiety here for targeted delivery into the site of action that you want. This will lead us to improve efficacy and, of course, reduce toxicity and improve efficacy. This slide is very important because this basically highlights the senses and the design and the assembly and the characterization of the nanostructure. So there are 4 key elements when you think about using nanotechnology. The material that you use to form the shell. You have to think about the size. What is the optimal size of the particles? You have to think about the shape. What is the optimal shape? And then you have to think about the surface area and the surface itself. You can do a lot of surface chemistry and surface modification if you want to achieve targetability and targeted delivery. Inside the nanoshell here, encapsulation of the active therapeutics. So if you talk about the material, there are many materials that can be used. And usually you have to think about a material that's FDA approved, safe, have no issues, because you are going to face that down the line. So you can use polymers, and I'm going to talk about those polymers, like how I just briefly here. You talk about polyglycolic lactic acid, a copolymer that's biodegradable, and that's have been used in a lot of applications. You can talk about natural polymer like chitosan from the shellfish or from mushroom, which is also biodegradable and inert. It doesn't have an adverse effect, FDA approved as well. You can make liposomes. You can use a hydrogel if you want to implant for sustained release. Sometime you can put, for example, testosterone for under the subcutaneous implant, and that will be delivered over 3 months. So you can have sustained release of the active therapeutics over extended duration. Metal particles, that the utility of the metal is, in terms of the safety profile is something that you have to watch for. So carbon nanotubes, this is also from human use, and putting it inside the human subject, you have to think about it. Dendrimer, you can do multiple branched particles, and then you can do protein drug conjugates for targeted delivery, and we're going to talk about that later on. So this is the material. A lot of material can be used, synthetic or natural, and there are a number of natural biomaterial that you can use here. The shape, sometimes in a way, the spheric shape is more preferred in terms of the delivery, and some other cases, the road shape, it can be preferred. So you have to look into the for targeted delivery, what shape would work better for you. The size usually between 1 nanometer to 100 nanometer to even 200 nanometer. Is because this will give you enough large surface area. And if you go 100 nanometer or 200 nanometer, you have enough space in the nanoshell to put more material in. So the loading of the API will be greater if you increase the size. But but you can use multiple nanoparticles even if you go down to 1 nanometer. And sometimes, lower nanometer, 1 to 10 nanometer, have a better profile. In other cases, you might need to go to 200 nanometers. So it depends upon the bioavailability and the efficacy. Surface is the key here. You can do pegylation of the coating on the surface to make the nanoshell, a nanoparticle, less immunogenic. And this is very important in many cases. You can do surface functionalization, for conjugation of targeted ligand. So sometimes you can, if you put an SH group or NH2 group or carboxylic group, that might help you to conjugate, a peptide or an aptamer or an antibody, or even a small targeted high affinity molecules. The charges sometimes could play a role, positive or negative or neutral. So depending upon the application that you want to go. So here is example of natural biopolymer. And if we look at the source of these natural biopolymer, they can come from plants, from animal source, from algae, microorganism. So you can see cellulose and certain gum arabic or gum, or starches. You can have chitin or chitosan, glycosaminoglycan, hyaluronic acid from animal source, shellfish, or even from plant like mushroom. Algae, you can have alginate. Alginate is another a good polymer that can be used. Microorganism, you can have dextran and many other forms. So there are a lot of nanoshell structure that you can either use it from synthetic source or from natural source. I just want to show you here many of the pharmaceuticals that we are benefiting from till now, and they survive for a long time. They came from natural source, nutraceuticals. For example, you have aspirin was driven from eucalyptus plant, warfarin from another plant, that was discovered in Wisconsin, and then digoxin, this was discovered by the ancient Egyptian, and for cardiac arrhythmia. And still till today, nobody, I was in pharma for 2 decades. We could not be able to replace digoxin. So digoxin, despite the fact digoxin has a narrow therapeutic window, but it is a very valuable drug. Heparin also comes from the animal source, from the intestine, derived from intestine of various animals, or the lung or the liver. And it's being extracted and processed from porcine, from camel. Dr. Warda actually did the early work on camel heparin with Dr. Lenhart. And so heparin is a very, very useful molecule here, and still actually being used after 100 years. Aspirin celebrated 200 years anniversary. Heparin, almost 200 years anniversary was celebrated. Taxol, from plant source. Statin from fungus source. So I'm just showing you here that a lot of good pharmaceuticals were driven from a natural source, and they actually stay with us for a long time. Many of the synthetic pharmaceuticals, they come and go, and they're easy to be replaced. But naturally driven pharmaceuticals, they stay, and they are very beneficial, and they are very effective. What happened also, some of the natural pharmaceutical was transitioned into a nanopharmaceutical like Taxol. Taxol was converted using an albumin as a polymer, and encapsulated inside the albumin as Abraxane. Abraxane, almost in Europe and the US, replaced Taxol because it is easy to be solubilized, and it doesn't cause irritation upon injection. Taxol, you have to solubilize it in an organic solvent, so it causes a lot of irritation upon IV injection. So Abraxane market is now exceed $1 billion worldwide. And the case can be made also that in the future, and actually it's going on now, that you can convert directly nutraceutical into nanopharmaceuticals via making nanonutraceuticals, and then it become nanopharmaceuticals. And instead of going through the nutraceutical pharmaceutical, and then you go to nanopharmaceuticals. So you can go directly through here. Drug discovery, as I said, I spent 2 decades, and I was very fortunate to actually be involved in the discovery of diagnostic and therapeutics, like Cardiolite, an imaging agent for cardiac scan, is still being used as a gold standard till today. And then you have short-acting beta blocker, osmolol. You have losartan as an antihypertensive. So actually, I was very fortunate, and also antiplatelet anticoagulants, like the anti-10A. So the discovery drug development, however, have a high risk. And it takes a long time from discovery to develop to even get the product in the market. So I did ask this question when I transitioned to academia 20 years ago. Could this nanotechnology shorten the time to drug discovery, drug development, and mitigate the risk and extend the product life cycle and building and intellectual properties, which is very, very important to secure funding and to attract the investor to put money if you have an IP. IP is very important. I just want to mention that because the key for academia to attract industry is to build the intellectual properties, very solid one, so you can get them to invest into your effort. And I, over the past 20 years in academia, I focused on building intellectual property and have a number of spin-off companies that are now in clinical development, nanopharmaceuticals, and another company is going toward the clinical development of a product that's now trying to get the IND. But so we have compound in phase 1. We have another one is IND going to phase 1. The phase 1 is going to phase 2 clinical trial in glioblastoma and several other cancers. So this is and no investor will put money unless you have a very solid intellectual properties. Just want to show you here to emphasize the percentage change in publication per year. And this is over 5 years from 2013 to 2018. And the green one here is the clinical focus area where nanotechnology and nanoparticle were employed. So you can see in vitro diagnostics, in particularly the sensors, the nanosensor technology and nanochips, you can see here relative to a clinical area of focus without nanotechnology, you can see a huge change, 200% increase in publications. Cancer imaging, you can see here the green one. You can see theranostic, where you can do diagnostic and imaging, therapeutic and imaging at the same time. So you see the green line. Combination therapy, because this is a good fit to actually put multiple molecules in the nanoshell. Immunotherapy, you can do targeted delivery of immunotherapy into the tumor itself, instead of having the cytokine storm that produce a lot of side effect to the patients. Gene therapy, it cannot happen without, if you have gene and you want to target do gene therapy, you have to use nanotechnology. Vaccine, you cannot do vaccine without protecting the antigen, which is very unstable. If you administer it as such, you want to sustain the antigen to stay for a long time in the circulation to build antibodies, and caging it and keeping it. And you can, actually, I'll show you an example of hepatitis B vaccine, which we were able to make it only single vaccination relative to 3 boosters every month or every 3 months. So even the Moderna and the Pfizer vaccine, really, if they didn't have enough time, they could actually make it a single injection, single boost, and instead of having to give 2 boosts. But the time was urgent for them to push it forward. So where nanotechnology, for example, I'm just going to use this case against the coronavirus, for example. But this can be applied to a lot of other infectious disease. In terms of, if you look at intellectual property, mask development, and there is an employment of nanotechnology, clothing, with virucidal activities using nanopolymer, and with antibacterial activities. You have vaccines. So the case is very clear. You can inhibit viral infection. You can also surface decontamination, decontaminate the surface by having a nanoparticle that can stick to surfaces and that actually prevent the survival of the bacteria or the virus, auto clean and so on. So you can see nanotechnology can help us in a way gain a lot of benefit for the society. I'm going to show you here, sorry, case studies, an application in the pharmaceutical field. You can actually use nanotechnology in drug discovery, drug development. You can shorten the time to develop and discover and mitigate the risk and extend the product life cycle in a way, which is an intellectual property buildup. A reformulation to improve pharmacokinetic and pharmacodynamic. And if you have an existing product, which will become generic, and if you can make an improvement using nanotechnology to make it more bioavailable and more effective, the FDA will give you an approval, as if this is new entities. So you can start the clock again, instead of becoming generic, where where you can actually now you can have a generic competition, but you have to have a significant improvement for the FDA to give you an approval. This is the case in pharmaceutical. I'm going to show you example, and vaccine, biopharma. What the more exciting here is the targeting. If you can target delivery instead of simply encapsulate inside the nanoshell to improve the delivery to a certain extent, if you can target the site of action to improve efficacy and safety, like chemotherapy, antimicrobial, dyslipidemia, I'm going to show you some of these examples, antiviral and vaccine. Show you here a reformulation of rapamycin by Wyeth Pharmaceutical, which was acquired by Pfizer. This is basically the nanoparticle suspension here. The drug was in the market and was given as as a solution. So now they want to make an oral formulation, and they want to use nanotechnology. So they created an oral formulation using nanotechnology, and they were able to improve bioavailability by about 27% and enable the preparation of tablet instead of actually suspension. And that get them an FDA approval for this product as basically a brand name product. It's called Rabamin tablets, and instead of using suspension. Another example here, Amend, which is a capsule now. It's based on nanoparticle suspension, and it's supposed to be antagonist. They were able to improve oral bioavailability and reduce. There was a lot of variation when they take the solution. The fed and fast variability in bioavailability, they were able to eliminate that using this oral formulation. And there are a lot of examples here that are already product in the marketplace where nanoparticles was used. And the FDA gave the approval because there was improvement in efficacy and safety, or the PK, or both, like the case here with this product here. Let's go to vaccine. How nanoparticle, nanotechnology can help us with vaccine. Here is an example which was done in our laboratory, hepatitis B vaccine. Here is the antigen without the titer, the antibody generated when you give the antigen alone. If you give the current vaccine, the vaccine that was available at that time, which is an antigen in alum antigen, this is the titer, the antibody generated. So the patient have to come up back again for 3 boosters. However, when we did the chitosan, which is a natural shell, we were able to put the antigen in a chitosan nanoshell, and look at the titer here. It was sustained and durable, where this will be only 1 single boost instead of getting this green one here, which was available in the marketplace, that you have to give it in 3 boosts. So that's a very quick example here in the vaccine. The Moderna and the Pfizer vaccine is another case where they used lipid nanoparticles. The other thing that we have done, this is the nano vaccine and nanotherapeutics. We were able to do a lot of work in a way with Dr. Lenhart and many other collaborators, where we found out that heparin in a way, and other low molecular weight heparin can effectively inhibit the coronavirus entry into the host cells. And also the heparin has high affinity to bind to skip protein, which is very important for the entry and permeation of the virus into the host cells. So we're able to formulate heparin or even heparan sulfate, which is non-anticoagulant, into a nano formulation and give it as a spray, as a by inhalation or as intranasal for to inhibit the coronavirus for prevention, and also for any residual virus that is in the upper respiratory system can be actually stopped from getting into the system. And we're able to combine this formulation with a direct antiviral compound that is actually was discovered way back against various coronaviruses. So it is this is patented work that we developed ourselves. The other thing I'm going to talk about, nano-active targeting, which is the most exciting, the most important for the future. How can we improve chemotherapy, antimicrobial, antiviral, dyslipidemia for lipid lowering? And so here is this is not an active nano-targeting, Abraxane. Abraxane received the FDA approval in 2005 for metastatic breast cancer. Now it's being used for various cancers. And it is, the content here is paclitaxel, albumin, and no surfactant, no solvent. If you look at paclitaxel itself, you have to solubilize it in organic solvent. This is very, irritating and very toxic to the patients. Here is very inert, and you can give it by IV, IV infusion without any irritations. And at the same time, it shows that you can get more into the tumor, but it's about maybe by 1 or 2-fold increase in tumor uptake. And this have no active targeting. If we look at the shape of the particles and the size of the particles is here, it is about 550 to 150 nanometer. And the hydrophobic drugs, paclitaxel, is included here. And this is the albumin. And this is a cryo-TEM that shows the particle itself. I'm going to just switch gears here toward a very active area in biopharmaceuticals. Antibody drug conjugate, EDC, for active targeting. So you can use an antibody, for example, against Herceptin, which is overexpressed in various tumors. And then you can make a linker with the antibody to be a payload here of a cytotoxic compound like paclitaxel or cisplatinum or many other chemotherapeutic agents here. And then the antibody, what the antibody would do, the antibody will target the tumor because it's overexpressed and deliver the payload. And then the payload will be actually, the cleavage here will happen in the cancer and the tumor microenvironment, and releasing the active cytotoxic compounds. So this is an example here. This can be also done in a nano fashion, where you can have the antibody drug conjugates decorating you can have the antibody directly conjugated to the nanoparticles, and then you can put the chemotherapy inside the nanoshell. So this will actually give you a payload with not only 1-to-1, because with the EDC, with the antibody drug conjugate, you have one antibody to one chemotherapy. But if you have a nanoshell, you can actually put multiple molecules of the chemotherapy, and then you can decorate the surface of the nanoparticles of the polymer of the nanoshell with multiple antibodies. So this is, we are also doing a lot of work in that area, trying to use, instead of antibody, use high affinity ligands, again, that for the receptor that's overexpressed, in that case, in the cancer cells. And then we can do conjugation with the chemotherapy here. Well-defined, very simple chemistry. And this is an area that we are actually pushing forward for a small molecule target ligands. And it should be, instead of dealing with biologics, this should be much cheaper and, and then, also, easy to reach to a lot of, so the stability here is not an issue because you're dealing with a very stable small molecule, high affinity ligands. So we are doing drug-drug conjugate. We are doing also, instead of using an antibody, we are using a small molecule, high affinity ligands, and load the nanoshell with multiple molecules of the chemotherapy for targeted delivery. So this is an area of very active research. We have a spin-off company from that effort here. It's called Target Thera Pharmaceuticals. So one of the area that we spend a lot of time in it, and we discovered that receptor that's overexpressed in tumor and various tumor. It is the integrin or thyrointegrin alpha V beta 3 receptors. And we developed a small molecule high affinity ligand for that receptors. We conjugated this high affinity ligands to a polymer, and then we made a nanoshell decorated with the high affinity ligands, and then we loaded with many different active therapeutics, including chemotherapy. And here is this is one of the area that we have done, and this was the beginning of this company that's now is in clinical trial in glioblastoma and other cancers. It's called Nanopharmaceuticals. So here is just an quick example to show you the delivery of cisplatin in bladder cancer. So if you look at cisplatin, if you put the cisplatin in the nanoshell like Abraxane, you can get about twofold greater delivery of cisplatin into the tumor. But if you put it in a targeted nanotetrac, which is the targeted delivery system here with cisplatin, you can get about fivefold greater delivery into the tumor, which means that you have less spillover into the normal organs. So usually cisplatin is associated with nephrotoxicity, kidney toxicity, so you can actually minimize or eliminate those toxicities of the chemotherapy. So another the same delivery system here. We put paclitaxel, and we were this is in pancreatic cancer. And if you look at paclitaxel delivery into the tumor, so you can see about 25 nanogram per gram tumors. A nanopaclitaxel, a very slight but not significant increase. But if you put paclitaxel with an active targeting, you can get tenfold greater delivery within that. And we were able to actually increase this delivery level, into the tumor by 20 folds. So, so this is just an example that you can actually put a molecular signature that recognize a virus or bacteria, which it doesn't exist on the human host cells, and develop a high affinity ligand for that, and then load it with antimicrobial. And then you can actually zap specifically the virus or the bacteria or the fungus in a way with the anti-infectious molecules. This lipidemia, this is a very quick overview. I can spend an hour on every one of these. So this is something that we spend a lot of time on it. There is an antibody that's developed that will bind to this enzyme called PCSK9. PCSK9 enzyme is overexpressed in the liver, and if it's expressed in the liver, usually it will degrade a receptor that's very important for taking cholesterol from the blood and getting rid of it. So people with high expression of this enzyme, they have a very high LDL cholesterol. And this is, nothing they can do. Even a statin doesn't work. So because this receptor, LDLR, will be degraded by the enzyme. So that, two companies developed an antibody, monoclonal antibody, and it's FDA approved already. And they can reduce LDL cholesterol and improve cardiovascular outcome. So this is very clear benefit. This is the two antibodies here, FDA approved for a number of years now. But the question we ask it, can we develop a small molecule orally bioavailable instead of giving people to get injection? And the injection have to be given twice a month, and it costs around $3,000 per shot. So this is, it's not for everyone. And even with the insurance companies here in the US, the copay is very high. You have to pay something like $300, for coverage, and the insurance will take care of the rest. But but you have to have that insurance. But people in the rest of the world, if they don't have insurance, they will be out of luck. They will not be able to get these molecules. So we said, okay, can we make a small molecule that can inhibit the interaction between this enzyme and the receptors, so we can preserve the receptors to be able to take care of high cholesterol in the blood and get rid of it in the liver. And indeed, we were able to show that. And here is the quick data. In preclinical models, this is a molecule was called it's a targeted, this molecule target the liver. It's a nanotechnology based, and a small molecule. And and this basically can go into the liver and reach out to the enzyme and receptor and prevent the interaction between them. And this will preserve the LDLR receptor to lower cholesterol. Here is the evidence. If you administer this oral, P21, this is after 1 day oral, and this is a different doses, and you can see LDL cholesterol reduction. You can reach about 60% reduction. If you look at the antibody, that's already FDA approved. 1 day, this is subcutaneous, this is oral. So you can actually achieve a comparable LDL cholesterol lowering with a once a day oral compound that can be given in a capsule. So this is just to show you how nanotechnology can make a major improvement and major development that can benefit humanity at large. The growth of nanotechnology and its application is on the rise. You can see here over the years, and we expect between 2020 and now there is a lot of mass market production and application of this. There are a lot of FDA approved product, and many other products in different applications are growing. So this is to show you in 1970 and up to 2005, there was a lot of basic research, applied research, and then we have an early adopter and then rapid advancement. But we started here to see that we are reaching a very significant number of products to be approved by the agencies. So so what also the other thing I want to mention here in terms of that nanotechnology and nanomedicine that will be developed using nanobiotechnology, they can lead us into precision medicine. And the precision medicine can help us to approach cure in terms of versus managing the disease. And nanomedicine can enable us to minimize because we can bypass the liver if we can target to the disease, the organ, so we don't have to deal with pharmacogenomics and in variability in pharmacogenomic blood levels due to pharmacokinetic variations, and hence pharmacodynamics, because you can go straight to target the delivery of the active therapeutics into the site of action. So you can personalize. You can actually do imaging, bioimaging, biosensor. You can use existing technology, boson emission tomography, MRI imaging, and you can functionalize a nanoparticle to give you a functional imaging versus anatomical imaging. You can improve definitely efficacy and safety. You can approach precision medicine and personalized medicine. And hopefully with the advancement in biotechnology, with gene therapy, and antigen development, for vaccine, for bacteria, for other diseases, and even for Parkinson's disease, for many other diseases, this will enable us to approach cure, because we can go straight to the target and and fix the problem using gene therapy to bring back the abnormalities, and get rid of the abnormality and the mechanism that's leading to the progression of the disease. So, this is the era of personalized medicine, and with nanotechnology, we can have an impact on gene, cell, and even an area that's really moving up here, digital therapy, where you can use both the digital technology in delivering therapy, and and eventually in the future, we think that even you can personalize where you have your name listed here on the therapy itself, because it's highly personalized to subgroups which have certain molecular defect that need to be fixed. With that, I thank you for your attention.
Thank you, Professor Shakir, for your comprehensive presentation. Let's cover like overview, like what you are doing and what are in the current era of medicine or treatment. So now we can just have like a few questions until like 5. I have a few questions for like I can just like look for the question from the audience. So now I have seen like most of the natural biopolymer that you already just mentioned is chitosan. But how about the others? This is from animals. How about, for example, from plant, from algae? So is there any like applicable like or any formulation that could, for example, starch, as you starch now is using everywhere right now, and they are trying right now to use a starch for biofilm everything. So is there any formulation or that are made from the starch or from algae like byproduct?
Yeah, allogenate. You have allogenate, allogenic acid, is being employed in some products. Chitosan is actually because it's biodegradable, and you can, we are using it for, actually, making heparin or, non-anticoagulant, even low molecular weight heparin for sickle cell disease, converting it from an injectable heparin have to be injected subcutaneous or intravenous, even the big heparin. So we were able, chitosan is a polycation and heparin is a polyanion. We were able to use chitosan to make this is IP protected patents, to develop an oral formulation using, this polyanionic, which cannot penetrate the GI, and using chitosan from natural source, from shellfish and from mushroom. And that will give us, a 40% oral bioavailability for treatment of vascular disorders. And in particular, the focus is sickle cell disease, but this also have various indications in a way to be developed in clinical trials. So the natural, I'm a believer in the natural products as a good source and safe, because it's natural.
Yes,
and I showed you a testimony that if you look at the pharmaceutical that survived, for 100, 200 years plus, and never been replaced with synthetic, man-made drugs, still with us. But those synthetic drugs, they stay till the patent expire, and then another company will come and modify those synthetic products, and they are out of the window.
Is there any report about, is there any resistance about this nanoparticle?
No, I think, what you have to do, you have to show, a safety. We have to do, for IND, for example, for us to get into clinical development, we have to do what's called GLP tox study in 2 species. And if you have safety margin, good safety margin, you can go into human trials. That's how we are doing it. And then if you can show For example, chemotherapy, you don't have to do phase 1, phase 2, you can do what's called bioequivalent studies. Let's say I have a chemotherapy that's already approved by the FDA, it's generic. If I put it inside a nanoshell targeted to the tumor, and I can show in head-to-head comparison in a bioequivalent studies, in a very short bioequivalent studies, that this is in a way superior, even equivalent, the FDA will give you an approval.
There is one question from the audience that they are saying that, are you using in silico studies?
You can use in silico studies to help, but in a way, and it is good, but you have to validate the in silico studies by doing an actual in a way this is simulation, which is good. It can actually maybe facilitate the discovery and development, but at the end of the day, you have to put it in the real test. In a cell-based assay or an animal experiments to be able to show, because you have to have PK and delivery, and so you can simulate the in silico is okay if it can guide us to determine the path forward. But you have to go to the real test in whole body animals, or in a cell-based assay, and then before you move it into human.
Thank you, Professor Shakir, for your nice presentation, and we are happy really to have you with us for this today's meeting that we always doing for continuous medical meeting, and we do hope to have you, inshallah, in the near future to have another talk about in details about personalized drug therapy and your area of interest. Thank you, Professor Shakir, and have a good day.
Thank you so much.
Thank you very much.
Thanks for the invitation.
Thank you very much for your presentation.
Have a good day.
Bye.
Bye-bye. Thanks so much. Bye-bye.
Thank you very much.